Hey, everybody, welcome to a very special episode of the Growing with Fishes podcast. This week we have the amazing Dr. Elaine Ingham joining us, one of the top experts on soil microbes and everything related to how soil interacts with plants. We're super, super excited. We've been trying to have her on for a long time and couldn't be happier. Thanks a lot for joining us, Dr. Ingham.
I'm glad to be here. This should be fun.
We also have Marty. He's joining us again after 2 weeks off. Thanks a lot for coming back, Marty. I guess he's AFK. We have Leighton from Kingdom Aquaponics with us.
Hey, buddy. How are you? Good to be back.
Yeah, been in a couple episodes.
Yes, it has.
And we got Fish Ganja Guy.
Pleasure to be here.
We got ECSD. And we have Josh Rutherford from Dutch Blooms.
Hey guys, glad to be back. Thanks for letting us all be on. It's fun to have these combos. Combo.
This weekend, we'll all be up at the Science of Regenerative Organic Cannabis Conference in Redway, California. If you're interested, definitely go check it out. Dr. Ingham has just finished teaching a 2-day course on mycology there, or not mycology, microscopy. I'm sorry, and yeah, just here to talk to us all about soil science. So thanks a lot for joining us, and please tell people a little bit about what you do and about the Soil Food Web.
I have a research farm that was bought for us by a funder who wanted to see the scientific replicated trials be done. We've worked a lot with individual growers, but of course, a grower's focus is on.
Wanted to see the. Sorry about that.
Focuses on trying to make money, really, being successful, keeping the farm together. So as soon as they started seeing how much easier it was, how much less expensive it is to use biology in the soil instead of toxic chemicals, pesticides, inorganic fertilizers, and all those things, pretty soon the experiment is gone and the whole farm has been turned over to the biological approach. So we could just never manage to get those replicated scientific trials for the year after year after year after year that we have to have for publication in the agricultural literature. So on the farm that's just outside of Oroville in California, we have started setting up trials. So we always have the control and the treatment. We're looking at without biology, with biology, and now we've added a comparison with the conventional toxic chemical approach. So we can be comparing our results. In the first year where we started putting the biology in on the surface of the soil, we increased yields with biology as compared to no added biology, same amount of nutrients added into the soil, but with the biology, we increased yields by 50%. In the second year, we increased yields in that same comparison by 300%, where we have added the biology deeper into the soil from the very beginning, breaking through the compaction layers, getting good structure. So water and air and the biology and the root systems of the plants would go down deep. We increased yields by 1,000%. So we want to keep showing that these kinds of yield increases will happen year after year after year. We clear cut a 3-acre area late last summer. And so we're going to be expanding the kinds of experiments that we're doing. So we're always coming up with new experiments. We have more things we want to work on than we have space to do it. So it's keeping us busy, keeping us in trouble, all those good things. So I still talk with people around the world. So I'm invited to come and give talks about the things that we've been doing, the success stories. You can go to our website, soilfoodweb.com, or for the experimental farm, it's environmentcelebration.com. And we put updates of what we're doing, updates of the soil life consultants that we're training and certifying. They have to go through a pretty good training course, so they prove that they know what they're doing and can successfully use the soil food web to get people off pesticides and inorganic fertilizers. So yeah, doing lots of things.
Very cool. I understand that you were one of the first people to figure out a universal way to assess soil microbes. Do you want to talk a little bit about that?
Yep. So the work with the microscope is what we need to learn so that you can very rapidly and very easily determine what biology is present in your soil. When we go into conventional agricultural fields, the only microorganism that's typically left is the lots of anaerobic bacteria, maybe a few of the bad guy fungi. Things that are not really going to help your plant grow, which is why you have to keep using inorganic fertilizers and pesticides and toxic chemicals to try to knock away all of the bad guy organisms that want to attack and eat your plant. We've been working with the microscope, and so we just finished that 2-day course where we were training people to use the microscope. You take a sample of your soil, so we went through how to sample soil, bring it into the microscope, make the dilutions, and in about 5 minutes you've got your sample ready to look at. And then we teach people what the appearance of the beneficial bacteria, the not so good bacteria, the beneficial fungi, actinobacteria, the bad guy fungi, the beneficial protozoa, the good and bad nematodes. So you very easily can see, do you have the set of biology required for the plant you're trying to grow? So yeah, I was the first person that really put together how we would be able to do all of these different kinds of organisms all with the same sample, and not having to take weeks and weeks to get the information back to people.
That's amazing. Actually, it's a question I'd love to learn a little bit more about. How do you tell a good from bad nematodes? Because there's almost always quite a few whenever you take any kind of sample.
You have to look at their mouth parts. You've got to look at the esophagus. It doesn't really take that long. I think pretty much everybody at the course in the last 2 days has the basics of what they need to be looking at. They need to practice now. They need to get the confidence that they really can tell the difference and they're going to be right every time. So it doesn't take long to learn these skills. And I think it's really important to be able to measure what you're trying to manage. How can you manage anything if you have nothing to measure it with? So we pretty quickly can train people how to use the microscope and look at the good versus the bad and know exactly what you need to be adding. You can look at your compost and determine whether you have the organisms in the compost that you're making that will put these beneficial organisms back into the soil. Compost is all about life. It's not about how much organic matter. It's not whether you put manure in there or whatever. All of those materials are going to be converted into foods for microorganisms. And so do you have the life in your compost that you need to put back into your soil? If you can't put a solid in, you can put a liquid in. And we teach people how to make the liquid forms of compost and apply them.
Awesome. That's so neat. There's so many different things that in that regard, and it's one of the biggest things out there. Personally, I've only had microbeorganics.com to use as any kind of reference. So it's good to hear that you have these great classes now that are much more in depth than the little bit that's out there online, and all in one condensed place. Are you teaching another one again soon at all, or have plans to?
We have online courses, so that if people want to sign up for the online courses, we give you a password that's good for an entire year. Because we know that people get busy. We know you're going to have to, especially with planting season coming up, you can get real busy for a couple months, and so then you go back to the online course, so you can watch it. Like a lot of people will do a 45-minute lecture each day. And pretty quickly you're going to get through that course. We have quizzes that you get to take to kind of test to see whether you picked up all the ideas, all the concepts or not. We then will take people who have passed those courses and train them how do you make compost, so some practical hands-on making compost extracts, making compost teas, and then do a project where you actually have to document that you're starting with dirt with no decent biology, and you're adding those organisms so that you're reaching the balances for the plant you want to grow. And we get rid of the pesticides, inorganic fertilizers, we reduce water use, you exit weeds, you don't have to deal with them anymore. So all these benefits that we can give people.
Awesome. Can you talk a little bit more on compost teas? That's definitely a topic. There's a lot of mixed information out on that. Do you want to talk a little bit about what makes a good compost tea versus a bad compost tea, and just maybe some of the myths out there as far as there's so much bad information, almost as much as good information on compost tea.
Yeah. You first of all have to have good compost, because we're going to be extracting those organisms from the compost into the liquid. And so you better have the organisms in the compost. I always get amazed by people who grab manure and extract the organisms out of the manure and into the water. And it's just like, oh, do you realize what you're extracting from manure? Those are human pathogens. Those are plant pathogens. Those are horrible things that you don't want to have anywhere near your plants. So you've got to start with a good material. You've got to learn how to make good compost. So then we're going to be extracting using water. We're going to add specific foods to encourage, stimulate the growth of those organisms that you're lacking in the soil. Now, if you've got a billion bacteria in your soil, why go in and grow lots more bacteria? It's not really going to help you a whole lot. So if what you're missing are the fungi, and almost everyone who's been growing in a conventional fashion, even organic growers where they still have a lot of weed problem, what you're missing is the fungal component. So you've got to put the foods into that compost tea to encourage the growth of those fungi. Well, bacteria and fungi in the soil grow on the exudates coming out of the root systems of plants. Those exudates from the plant tell the bacteria and fungi what enzymes to make to go out into the soil and pull the nutrients from the sand, from the silt, the clays, the rocks, the pebbles, and of course organic matter as well if you have them. But when we start to understand that the bacteria and fungi pull these nutrients from all of this mineral material, and it can't be soil if you don't have those mineral materials in the soil. There is no soil on this planet that lacks the nutrients to grow plants. So why are we putting out fertilizers of any kind? Your soils contain thousands of years worth of the nutrients that your plants require. They're not in a plant available form. So we have to have this biology in the soil in order to convert what's not plant available, but which is there in huge quantities, convert it into a form that your plant requires. So that your plant can take up, So the bacteria and fungi are pulling those nutrients into their bodies, and they're right around the root system, and they are basically storing those nutrients that the plant wants. And so the protozoa, the nematodes, the microarthropods, the earthworms come into that root system because there's lots and lots of fungi and lots of bacteria. And those predators eat the bacteria and fungi. And because the nutrient concentration that the predators require is so much less than the nutrient content in the bacteria and fungi, they're going to get rid of those excess nutrients. And of course, they're going to release them in a plant available form. So if you ever want to get away from having to use fertilizers of any kind, you need to get this biology back into your soil. We want to make certain that we're putting out those microorganisms that are beneficial so that weeds will not be able to grow. We select against weeds and we select for the plants that you want. We're going to make certain that we are protecting our root systems and the plant material above ground. with the biology that is going to be protective and makes certain that there's no space, there's no food, there's no room for the disease-causing organisms, for the pests, for the problem organisms. So when we're working with the compostee, we want to make certain that we're getting these organisms up to these high concentrations. So when we spray on the foliage, we are covering that leaf surface, those leaf surfaces, top and bottom. We're covering the branches, we're covering the bark, we're covering the fruit, the flowers, whatever, so that you have these highly beneficial organisms that are going to make certain that you don't have diseases and pests above ground. We can be putting compost extracts, where what we're trying to do with the extract is just extract the organisms out of the soil. We're not adding foods into that extract. We're just extracting the organisms. We're going to extract a bit more compost so that we're getting the organisms up to the really high levels that we need. And then we'll be applying the extracts into the soil. So if you can't cheaply or easily apply the solid, you can apply the liquid forms in the soil or on the foliage and protect your plant, give your plants improved nutrient cycling, get the root systems of your plants growing as deep as possible, getting really good water infiltration into the soil. So in places, for example, like Australia, we have done studies where we've shown that if we build better structure in the soil, get rid of the compaction layers, we can reduce water use by those plants by as much as 50 to 70% in the first growing season as we're putting this biology into the soil.
That's amazing.
Yep. And of course, in some place like California, that's hugely important. We have to reduce our consumption of water. We have to not have all the toxic chemicals and the pesticides leaching and washing into our groundwaters and into our surface waters. Yeah, I always have to kind of laugh when Monsanto says we have to have these genetically engineered organisms in order to feed the world. In 2025, we're going to run out of food to feed people. Well, guys, that's absolutely not the case. What's going to wipe people out long before 2025, when we're going to apparently run out of food to feed the planet, is water. There's no clean water. Right now, most people are suffering from a lack of clean water to drink. What's going to kill you? It's the toxics in the water, not the lack of food. That may or may not actually be true, that we're going to run out of food to feed everybody by that time. We've got more than enough food to feed everybody. We just have a really bad time getting that food to the people who are starving to death.
Is there certain compost teas or anything to help breed those predators of fungi and bacteria?
Yes, so you have to have high concentrations of bacteria so that the bacterial predators have lots to eat. We've got to have high levels of fungi so the fungal predators have lots to eat. So it goes to that base of the food web. We've got to grow lots of these first trophic level things, so we can have lots of the second trophic level things. We can release nutrients at the rate that the plant requires. And actually, it's your plant that determines how fast this is going to be happening in the soil. If your plant is healthy, it's got all the nutrients that it requires, the plant can be making the photosynthate in order to supply all of those exudates to release into the soil and get the bacteria and fungi to do the job that the plant wants to have done. So, we as human beings don't know what the plant requires. The plant knows what it needs. So rely on that communication between the plant telling the bacteria and fungi what to do. And the bacteria and fungi are only too happy to go do that because they're getting fed. So they do the work the plant requires. You and I don't have to know that what your plant needs right now is a couple micrograms of boron. But as soon as your plant gets that boron, now it's going to need something else. Something else is limiting. So now it needs some more calcium. When the plant gets the calcium it needs, now it's going to need more nitrogen, and then it needs more phosphorus, and then it needs, on and on and on. Every second of every day, your plant is got to be given the nutrients that it requires so it can stay healthy. And of course, if your plant is staying healthy, then pesticides, inorganic fertilizers, toxic chemicals are not needed to get rid of the pests or the diseases. If we're making certain that we have ammonium in our soil instead of just strictly nitrate, we select against the weeds, and we select for the understory plants that we need to have in those systems.
So with that being said, would you say that Mulder's chart is still an accurate way to help diagnose deficiencies or balance things, or do you think that that's kind of the wrong way to think about things?
Well, when you're looking at your plant, you can discern when there's a really critical shortage of something. So the leaves, the color of that can tell you that your plant is just about ready to die. But we want to maintain that nutrient uptake by the plant and make sure it's properly balanced long before there's any deficiency that shows up where you and I can see it in the plant as yellowing veins or drying tips of the leaf or purpling that your plant is sick unto death when you start seeing those signs. So long before that, we want to be solving any nutrient deficiencies. So our job just needs to be put all the different species of bacteria and fungi you possibly can into that soil, and the plant will feed what it needs. We need to have the microscope. So we take soil samples and we make certain that the bacteria and fungi, protozoa, nematodes are all in the proper balances, which means the plant will be getting the nutrients that it requires. There's our testing ability. We need to be pulling soils and taking a look at whether we have that biology in the soil. We need to take leaves off the surface of the plant and use the microscope to determine whether we've got the coverage on that leaf surface by these beneficial organisms, so there is no way that diseases and pests can bridge that castle wall, protecting those leaves, the stems, the fruit, the buds, the flowers.
Very, very cool. Have you worked at all with any of like the Korean natural farming stuff, any of the fermentation, or experimented with any of that? And can you talk on that a little bit? That's definitely an area that I've been doing a ton of research and work with, and trying to separate different microbes from that, especially with kombuchas and a couple of others. And I'd love to hear about any of your work with that and kind of your research around that.
Yeah. What we see is that it's okay to have kind of these the best terminology is facultative anaerobes. They have the ability to live in aerobic environments and in anaerobic environments. So if you've got really bad compaction in your soil, you've got places where you've got to break up that compaction, and you've got to have things that will be able to grow in those reduced oxygen conditions. Then it's really useful to put in some of these lactobacillus and some perhaps some of the bacillus species, pseudomonads, that are capable of growing both in anaerobic conditions as well as aerobic. But the job of those fermentation organisms really is to start to build the structure. But as soon as you get really good oxygen moving into that soil, those organisms are no longer capable of outcompeting the aerobic organisms. So now we want to be adding those aerobic organisms so we maintain that nutrient cycling that's needed by our plants. So the Korean natural farming is really, in my mind, it's a first step of moving into healthier soil. I then want to make certain that we're getting that whole food web that's going to operate where we have no reduced oxygen conditions in that soil, so our roots can grow as deep as they possibly can. Nutrient cycling will be happening all the time. We won't have weeds. We won't have diseases or pests.
I know it's certain. There's some research out about as far as certain lactobacillus increasing CBG and certain other precursors. I know you've done a lot of work with researching THC and CBD, as far as which microbes and stuff like that, and that's some of the cooler work. Do you want to talk about that to the extent that you're wanting to? That's one of the cooler things that I've been doing, and we've done some research ourselves with aquaponics and noticed that when we run the split root zone, the half in soil and the half in the flood and drain aquatic, we're noticing almost 100% of the time an increase in CBD across the board, and as well as THCV in particular. And we're still studying it, but we're attributing it to something to do with the microbes in the soil layer, because that's the only thing we can attribute to as far as input wise. So I'd love to hear your work on that.
Yeah, what we're seeing is that if we have a very bacterial dominated soil, we tend to get a whole lot more of the THC. As we start to shift and we get more fungal biomass in that soil, we start to shift towards more of the cannabinoids. So, so the CBDs that we want. As we deal with different cultivars of cannabis, there's different responses in each of those species about where that shift from bacterial dominated to fungal dominated, and what kinds of chemical responses we're getting in the plants. So we're seeing that it's very much that shift from bacterial dominated to fungal dominated. And of course, as you're shifting from bacterial dominated, the bacterial predators are way more important. As you shift towards fungal dominated, then the fungal dominated predators get a lot more important. So, we need to continue looking at what is the biology exactly in these different conditions where we've got roots split between the soil and the aquaponics, where you put in a lactobacillus species. What's really going on? What's the lactobacillus doing? What are the fungi doing? And exactly how are they interacting with each other? And the only way we can really try to understand this is to use our microscopes and see if these, when you add the lactobacillus, how long do they survive? What are they actually doing? Are they attracting more of the fungal predators or more of the bacterial predators into that root system? Just so many questions on what the mechanism is. And if we want to have different kinds of CBDs, we want more of, this one or that one, how do we manage, and can we use the biology to do that increase in specific kinds of compounds in the buds?
Have you done any work with as far as using specific bacterial cultures or even composted cultures or isolated cultures, and then using that to isolate certain plant enzymes or proteins? That's some of the other interesting work I've seen from not only the stuff that we've done, but a couple of other people, and something that I've been dying to ask you.
We haven't really had the opportunity to do that. We don't have the funding to go after those kinds of questions. So I would love to hook up with people who can do that kind of testing, where they've got the facilities to assess exactly what is being improved in the leaf material, in the buds. So yeah, let's hook up and work together. I'm not somebody who wants to say, well, I'm the only person knows how to do this, and you're gonna have, there's lots of people out there we've been training to do the microscope work, and let's all work together on getting this figured out as rapidly as possible.
So what types of inputs can people use that will, what's the word I'm looking for, encourage a more bacterial dominated soil versus a fungally dominated soil?
So you want to be putting in fungal foods or bacterial foods. So bacteria like very simple sugars. Their enzymes are designed to go after very simple compounds. They're not highly branched, not a lot of very structurally complex kinds of structures within that material. So bacteria are very good at the simple stuff. Fungi are good at the more complex. When you start to look at carbon to nitrogen ratios in plant material. The bacteria are going to use the very narrow carbon and nitrogen ratio materials. Fungi are going to do way better on the wider carbon to nitrogen ratio materials. So bacteria do really well at decomposing the carbon nitrogen ratio materials that are at 10 to 1, 20 to 1, 30 to 1, maybe up to 60 to 1. But when we start getting into that 50 to 1 ratio, carbon and nitrogen ratio, now a lot of the fungi are going to win. We get into 100 to 1 or 150 to 1 carbon nitrogen ratio material, the fungi are the rulers there. So as we try to shift things into a more fungal dominated soil, we want to make certain that the foods we're putting in there will select for the fungi. And of course, the exudates put out by plants are gonna, they use exactly the same approach. If these are perennial plants, they're putting out way more complex exudates into their root systems. They make woody plant material, and that of course drives the fungal biomass. You deal with early annuals. The exudates are very simple. They're selecting for the bacteria. So this whole move towards understand the biology, what do you want? That's what we're going to use, is the foods in the compost, the foods in the compost tea.
Do you have any recommendations on companion plants or plants that would help?
Yeah, absolutely. We really, Mother Nature abhors bare soil. We never want to have bare soil in any of our ecosystems, because if that's bare soil, Mother Nature is going to put a plant in it. And of course, she's going to choose to put the plants that like that highly disturbed bare soil situation. So to make our lives easier for us, we should be planting the specific species of plants, short, low-growing plants, so they're not interfering in any way with the plant of our choice. What's the crop you want to grow? Let's make sure that these things don't interfere with that. So short, low-growing, perennial. I think is absolutely amazing in agricultural systems when people start putting in annual cover crops, because you're going to have to try to whack them down, turn them in, disturb the soil, cause problems with respect to oxygen getting into that soil when you have massive quantities of residue that you're putting down on the soil surface. And then you're going to have to buy seed again in a year to go back through all of this craziness. So let's put short, low-growing understory plants that have exactly the same mycorrhizal fungi that our crop plant, that our cannabis has. So let's match up these requirements below ground. So when we're looking at plants, understory plants that really help plants that like things a little bit more on the fungal dominated side, we want to be putting in woodier types of understory plants, like say creeping thyme or creeping lavenders, some of the isotomas. Where we're dealing with a little bit more on the bacterial side, let's be putting in things like dichondra, or some of the short, low-growing clovers, or a number of different plants. We have a number of these different plants, understory plants, cover plants, if you want, listed on our website, and we are testing those different species at the experimental farm just outside of Oroville, California.
So would that be like, along with the bacteria to fungi ratio,
exactly,
maybe planting accordingly on those cover crops, or like do we shoot for a mix, 50-50 or 60-40, 70, like how do we plant that out in a like a no-till bed, like where I have an 8-foot wide by 90-foot wide bed, and I want to
Yeah, this is where we've got to do some of the research first. Grow the plants by themselves, or by themselves with your desired crop plant. We find out whether it's really a good match or not. Are they really sharing mycorrhizal fungi? Are they supporting each other's growth? So true companion plants. And I always had to giggle when with the permaculture guys. They will say, in Australia, these two plants are companion plants. Well, great, that's in Australia. They're not companion plants here in the United States. So quit beating your head into a wall trying to force them to grow together better. So we need to be understanding what the requirements of the cover plants and the requirements of our crop plants actually are. And I want to see a cover plant promote the balance of fungi to bacteria, the protozoa, nematodes, microarthropods, earthworms that our crop plant requires. So there's a bit of research. We've just been starting on this for the last 4 years, and we've got a ways to go. So the more people who contribute and do some of this testing, the faster we're going to be able to get this information out to everybody on how to prevent weeds, how to promote the growth of your crop plant, and make it even easier for us to plant everything in the springtime and then walk away and we come back and harvest. That's how farming should be done. That's how plant production should be done.
That's so wonderful, and such a simplified explanation of all that. It's often people get it's hard to grasp sometimes. Do you want to talk about some of the work you're working on now at the research facility and some of the neat things you're working on right now?
Yeah, we're doing quite a bit with having. Well, the reason we bought the farm up just outside of Oroville, it's actually up by Bear Creek. The reason we bought that farm is the people who had farmed there before us were absolutely toxic chemical fanatics. They had applied so much Roundup and so much herbicide, fertilizers and things to that soil that in quite a bit of the area that there was their garden where they were growing food, couldn't grow anything anymore. Not even weeds would grow in the certain areas on the land. And that was perfect for us because then we didn't have to go in and try to kill off all of the biology in the soil. We didn't have to do anything to make it horrible dirt. So that became our control areas where we would take that field that had been completely denuded and you couldn't even get weeds to grow. And then we started with leaving half of it in the same condition. We didn't put anything in except some mulch to put in the same amount of nutrients, but not the biology. And then the other half of the field, we put in compost. The first experiments that we really put in were to only put the compost in on the surface of the soil. And then, of course, when we added the liquid forms of compost, the compost extracts or the compost teas, the compost extracts went on the soil surface. The compost teas went on the foliage to protect the foliage. So in doing that comparison in the first year, we documented where the compaction layers were in the soil. And we had a compaction layer right up at the surface of the soil, compaction layer at about 4 to 6 inches, another compaction layer just below that at like 8 to 10 inches, another compaction layer just below that. It was just kind of a story of, oh my gosh, how could any roots ever get through the surface of the soil and grow as deep as the roots of these plants can grow? So the compost put in on the top, the compost extracts going in on the surface of the soil in that first year only formed structure in that soil down to about an inch, maybe an inch and a half. And so, not a lot of ability to start to rebuild structure in the soil and do the job that we want to see. So in that first year, however, where we had the plus biology, the plants grew, we had a 50% increase in yield. In the second year, just putting the compost and the compost teas on the surface, the root systems got down about 3 inches. We will be seeing how much further those root systems got in the soil. For in this growing season, we'll be looking at that as well. So as the root systems go deeper and deeper and deeper, as we get more nutrient cycling around the root systems of the plants going deeper, our expectation is we're going to continue to see ever increasing amounts of yield because those plants are going to get healthier and healthier. Where's the maximum? How much increase in production can we get to? How much improvement in nutrient concentration in the food that we eat is going to be possible? So those are, some of the things we've been seeing in the last couple of years. And then in the area where we, right from the beginning, tilled the biology into the soil. And yes, I know tillage is horrible, but when you've got a compaction layer at 1 inch or 2, 1.5 inches, and you've got another one just below, and another one below that, and another one below that, how do you break up that compaction if the microorganisms can't move in very rapidly from the surface? So you're going to go ahead and till one last time. So we put compost on the surface of the soil, and we went ahead and tilled it into the soil so that we could get it down as deep as possible. Another approach that we're using is using like keyline plows or deep rippers. And on the back sides of those tines, of the stanchions going down into the soil, we run PVC pipes where we will be spraying out compost extract into the soil on the back side of those rippers. So inoculating all of that soil where the compaction is being broken up, where we're getting cracks and crevices formed in the soil, and allowing all that good biology to move into the soil. And when we do that, we're seeing increases in yields that are far greater than what we were seeing where we were just putting the biology on the soil's surface. This last year, one of the things we were doing with starts is looking at potting mixes that were prepared using chemical commercial potting mix that had toxics. We were supposed to put on, inorganic fertilizers, just following the whole regime of the chemical world. Versus where we put in just the biology into the potting mixes. So we were comparing those two results. And every time the plants, the seeds put into the plus biology treatment germinated and started to grow at least half the time it took for the seeds in the chemical system or no added biology to germinate and start to grow. So you're going to get a full, week more of growth where you're using the proper biology in the soil that you're planting into. And of course, that carries through the whole life of that plant. When we have pictures that we will, when I get back to the farm and have the chance to really sit down with all of the data, we will be putting together the report on that particular experiment and showing that you germinate in the plus biology, the seed germinates in half the time. And of course, then your first full leaf is going to be growing on that plant, a full week or 10 days sooner than in the chemical plants or in the no biology plants. We're getting, the height of those plants is double or triple the height of the seedlings coming out from the soils where there was no decent nutrient cycling going on. Here's what the biology is doing to you. It's very important. As I said, we're just clear cutted in whole new areas. So we're going to be putting in an apple orchard, understory plants. We will have, half of all of the plants we're going to put into the new area will be continued. We will continue to compare between the no biology the chemical commercial approach, and then the plus biology. We're doing some interesting work. We're going to be growing freshwater shrimp in our pond, and so trying to do that by growing all the foods that those shrimp would normally be eating in the real world, and making certain that we're maintaining that biological system. So it's predator-prey interactions, instead of feeding, the pelletized fish meal, or, the commercial chemical food that you would normally feed in these aquatic systems. So that's going to be a challenge to get that pond into shape and be able to do all of the testing to watch and see how fast are the shrimp growing, what's the effect on the pond. In most commercial productions, that pond water goes terribly anaerobic, and it's highly detrimental to the health of the shrimp. So we want to start those kinds of experiments. We have a food forest that we're going to be putting in. So we've got the trail built into the site. We will be putting in the terraces this year. We have light gaps, so this is truly a forest. This is not an orchard. We're not planting orchard trees and calling it a food forest. This is an actual forest, and we're going to be using the light gaps in that forest, making certain that we're putting the right biology around the root systems of all of the herbs and the ground cherry, the shrubs, the trees. We want to grow a whole set of different things. I have a friend who is going to be bringing me tea bushes from China. A bunch of different trees and a lot of different plants that we want to put in. We'd like to try coffee. We want to grow coffee and see if we can't do that.
Booyah, booyah. Oh, you just made my day. We got to talk about that. Serious.
I like the idea of planting and finding the plants in the forest and not having a true orchard, but using them in the space and open spaces. I've thought about that a few times, and I've got a kind of a forest out behind me. I'm looking forward to seeing what happens. Maybe I'll do some research myself.
Yeah.
Hey, wait about the shrimp guys. What about the aquatic ecosystem?
Right.
That's what these
Come on, guys. This is Aquaponics show, and we're talking aquaponic systems, aquaculture.
I do have to cut in just to be respectful. Elaine's been working all day, and so we got to cut to one more quick question and give her some peace and quiet tonight. It's so kind of her to come on after all of her hard work. Thank you so much, Elaine.
Thank you.
So one more question, and the shrimp. We can talk about the shrimp, whatever, but just we got to get off the air with her. So
sure. I had one that quick question in regards to soil. Is there any soil amendments or anything like that you'd recommend for people, or ones you recommend avoiding altogether? Or, there's so much information on that.
Yeah, I would want to have people test and make sure that any amendment that they're putting into their soil isn't killing the biology in their soil. And it's so easy to answer that question. You take some of your soil, you look at the biology that's there, add the amendment in the appropriate concentration, and in 2 or 3 days you look at your biology in that material again, and it's like, yeah, everything's growing. The fungi are growing better. The bacteria are growing better. The protozoa and nematodes wow, we're going to be growing great stuff because the nutrient cycling's going. Or you come back to that soil after 2 or 3, 4 days, and you look at it through the microscope, and you go, oh my gosh, what's happened to the protozoa? All of the nematodes are dead. They've been completely wiped out. The fungi are completely gone, and the bacteria are just taking over everything. It kind of smells bad. Well, this is not going to help grow your plant.
Great. Well, thanks a lot for joining us. Do you want to mention your websites again too? I have the Soil Food Web website on there, and then you talked about a couple of other references on where you're posting your work. Do you want to go over that again for people so they can find out more about what you do?
Yep. So my personal website is soilfoodweb.com. But for the research farm, the website for that and for the training program that we're doing for soil life consultants is environmentcelebration.com. So go take a look at both of those. And on both of those websites, we have a lot of reference material, a lot of the papers that have been written about compost and what real compost is. Compost is all about life. It's not about the manure and straw. You can't make good compost with manure and straw, and some of the conditions that have to be met. So lots and lots of research papers on compost extract, compost teas. I do try to point out that probably 50% of the papers in the scientific literature about compost are just 100% wrong. They're incorrect. They're not paying attention to what compost really is. They go out and they buy some municipal organic matter that's been sitting around in a pile for a couple years, and they use that to put on their plants or use as potting mix, and all the plants die. And so the paper says, we've proven that compost is really dangerous. It's going to kill your plant. Well, they never asked what compost is. They never established that what they bought from the municipal composting operation was actually compost. They never took any assessment of the biology in that compost. They might have done a plate count, which tells you about human pathogens, but we should never have human pathogens in a properly made compost pile. So you got to actually test the compost and make sure that it is compost. If you're going to be working with a compost extract, to make certain that you're using compost. I always have to laugh about a researcher at Washington State University who'd heard one of my talks and was just really ticked off at listening to, like, I didn't know what I was talking about or something. So she goes home and she grabs a couple handfuls of manure and throws it into a 5-gallon bucket and fills it up with water and swishes it around for 5 minutes in this direction and then reverses and goes 5 minutes in the other direction. And yes, that is aerating things. That is, getting some oxygen back in there, but she only did it for an hour. It's, the kind of the biodynamic approach. And then, of course, she left this bucket of manure water that she was now calling compost tea without any compost in it. There was no compost in that bucket. She leaves it overnight in her kitchen. I can't imagine what her kitchen smelled like the next morning with a bucket of manure water hanging out in there. Then she takes that bucket out, she fills up her sprayer, and she sprays her cherry tree with that manure water, and she killed her cherry tree. And so she writes a column for the local garden store, and what she writes is, I have proven that compost tea will kill your plants. And just, geez. And she calls herself a scientist? My gosh. It's just incredible, the literature you can sometimes come up with in the scientific world is just unbelievable. So when you run across papers that are just that equally as ridiculous, send them to me and I'll put them up on our website. See, even scientists can be unscientific. So we have a lot of references that people can use to show that compost tea, compost extract are perfectly capable of preventing diseases and preventing diseases and pests from attacking the plant, get rid of weeds, all the things I've mentioned so far.
Maybe quickly plug the soil life consultant thing, because I think there's definitely an audience here that would be interested in that.
Yeah, so we have certification program that we have. So we ask that people first take all of the online courses, and you learn the theory, you learn the practical approaches to making compost and compost teas, compost extracts, using the microscope. So you can now start doing samples on all of your land. You could maybe even start doing samples for your neighbors and make a little money at helping other people understand what's going on in their soils and how to improve their soil with the proper biology. We then want, in the for certification, we want you to actually make good compost. We want you to make really good compost extracts, compost teas, apply them, document that you know what you're doing. And then we ask that you do a project where you have to take dirt, and you first of all have to prove that it's dirt, and that's based on what the biology is. Just bacteria, just anaerobic organisms, nobody good home. And you've got to add the biology to that dirt and convert that dirt into soil. And so your success story goes up on our websites, and we start making certain that you are going to get clients, because we want all of our certified students to be successful and spread this knowledge and help everybody else reach this goal of getting off the toxic chemicals, keeping the money that growers are working so hard to sell their crops and get the money that they need to stay alive. And that money should stay in the growers' pockets, not be given to companies that promote things like genetically engineered organisms or toxic chemicals or pesticides or inorganic fertilizers that are killing us, destroying water quality, not making quality food for us to eat. So I thank you for letting me have the time to plug our program. So looking forward to interacting and talking with everybody further.
Very interesting. Fun too.
Thank you so much. And if you're there tomorrow, I'll see you tomorrow in person. And if not, I'd love to have you on the show again. I know you're a little time constrained, but thank you so much for taking an hour to spend with us, and we'd love to have you on again. I'm ecstatic about you coming on the show today. Thank you so much again.
Thank you. I'm glad to be here.
Thanks a lot.
Awesome.
All right, Steve. Hey, thanks so much. I'll see you tomorrow night. And Josh, great to keep it dialed down. Yeah, we got to get Elaine some dinner and get her to bed. She's cooked. And Elaine, okay. Great job, man. That was beautiful. You spat it all out. Perfect.
Thank you so much. I really appreciate the chance to talk to everybody.
Thank you.
Good night, all.
Ciao.
Awesome.
Hey, thanks, guys. I'm going to hop off too, but sorry to be so quick and brief. She really has been working really hard with. We had 25 students today, including myself. So she's been working really hard since about 8:30 this morning. And yeah, so I'm going to hop off too. I've been also doing it. So thanks guys for the platform. And I know she's going to really appreciate it. So.
Yep. And Josh, do you want to plug the show starts tomorrow?
Sure. Yeah. Hopefully I don't cut out this time again. My battery's plugged in. It's having like 2 or 3 weeks in a row. Yeah. So we're starting tomorrow morning at 9 a.m. and we're doing the Science of Organic Regenerative Cannabis Conference in Humboldt, in Redway, California. And it's a 3-day event. And we're starting with Layton, who was just on, and he's going to walk us through it. We kind of like have it staged out where we're walking through, the beginning of microbes and how they affect us. And then we get into mycology with Efren, who was on last week. And then also Chip, who we're going to dinner with tonight, Osborne, and he does some really interesting work. And then we just kind of work through the succession of how all this works together. And we have,
Chip is the KNF guy, right?
No, Chip is the guy who does all the soil food web on large scale, like big parks for where children are present, and they don't want toxic chemicals sprayed. So they want to do organics, but they also want it perfect all the time. So he has to be spot on, and so you get a guy talking like that to us. That's the same thing we need. We need to be spot on all the time and safe. So. And then again, Suzanne Wainwright, who's an entomologist, and she's really pushing it and challenging a lot of stuff and making it cheaper for us to buy beneficials. And then Chris Trump, who is the KNF guy, and he might be interested to come on. We'll talk to him when we meet him. I've never met him, and but I'm really excited on that because he does. I think it's like 800 acres of nuts in Hawaii, KNF. And that's a serious deal, serious investment, and it's pretty exciting for me to be a part of this stuff, you guys. To be honest, like there's a guy who's here for the class who runs cattle on. They have 800 acres they own, and like 64,000 acres of government land they run their cattle on in Idaho. 64,000 acres, and they're trying to convert it to all soil food webs. They're injecting and putting compost extracts and stuff, and it's crazy, man. I'm sorry, long day, but it's been really interesting stuff, and the conference goes on, and then it's going to be a lot more of the same. So if you guys are around the area, please come.
Awesome. Thanks again.
Yeah,
Thanks,
guys.
Josh.
Have a good night.
Cool. And check it there. All right. Cool. So that was really, really awesome talk, and hopefully we can get her on again too sometime for another talk. That was really, really cool.
Did you start early tonight?
Yeah, it sent an email out.
Oh, I didn't get it. Okay. I went. Okay. I was checking for a response, and the last thing I saw was Marty. So when I came up, my wife had emergency extractions today, so I was trying to make her some food when the show was starting. Dadgummit, I missed the beginning of the show. Can't wait to watch it.
Hey, Steve, can you hear me?
Yep.
Steve, I just want to plug Chip. I think you need him on this program as well. It's Chip Osborne at Osborne Organics. He does the chemistry side. He does exchange sites, cations, anions, how different chemicals work together to steal electrons, bind stuff up, release stuff. So I'll talk to him at dinner tonight about getting in touch with you. You'll meet him tomorrow again. But I think he's the other key piece to this. You got the biology, you got the chemistry, you got the soil structure. It's the complete package. So let's talk tomorrow when you get up here. All right.
Awesome. I feel like we learned about all kinds of new rabbit holes today.
Oh, dude, these are rabbit holes that go. Awesome. All right, brother. I gotta go take these guys out to dinner. So.
Do you want to plug what you do?
Oh, I'm a very humble guy. I'm just lucky to be a part of, all of these people. We're all out here sharing the love so that all you guys can get connected to the right approaches to things. So, I do Kingdom Aquaponics.
Did we lose him?
Sounds like it. I always have to wonder whether we lost him or we froze up. Never know. So I move around real quick.
We heard him at Kingdom Aquaponics, and then he cut out. All right. Well,
I don't know. Did I get shut off? I did. Yeah. So I'm about getting you guys inoculums to get you going and teaching you how to do it on your own. I have the worst business model in the world. It's about, teaching you how to do what I do and then not making you a client.
Kudos. Join the club. Yeah.
All right, guys. Josh is busting my phone up. Elaine needs to go to dinner. So I'll catch up with you guys soon. And yeah, I'll try to join some more podcasts. But when I'm out on the East Coast, I'm usually asleep by now because I get up early. So bear with me. All right. Good night, guys. God bless you all. All right. Godspeed.
Yep.
Good night,
Thanks
brother.
for joining us.
All right. Bye.
Cheers. All righty.
Marty's got 2 videos up here.
Yeah. What have you been up to, Roger?
Oh, well, just taking care of Mama. We're doing some upgrades on the forum. We've got a lot of new genetics that we procured, and we started selling some mix packs. So I've basically been doing marketing, helping out, and going to the Discourse forum, which is the forum for the software we use for our websites. And trying to learn to code again. I gave it up 5 years ago when I went blind, but I've got so much work to do that me and one of the administrators in the office in Amsterdam and I are working on upgrading the form, making it look better, adding some more user-friendly things. And we're always trying to figure out a way to spread the word, along the social. Other than that, trying to catch up with around here, waiting for the last frost to come. Hopefully it'll be pretty soon, because I want to try to get the greenhouse going again this year after a couple of years being dormant. And I got to go cut down all them blackberries to make some concentrate via Marty and Brain Grows, kind of ideas. And I can't wait to do that. It's just so much stuff and so many different things. It's hard to get to anything sometimes, Like I got 2 no-dig grow beds out there that are just growing over with grass and weeds right now because I hadn't had time to attend to them either. But now that we've had somebody talking about those, I'm sorry that is an aquaponics show originally, but we've kind of expanded that. But talking about the soils and the no-dig grow beds and all, I found that very interesting because I've already been experimenting for 2 or 3 years on that. And I've got, like you said, there's all kind of like rabbit holes or wormholes or, little crevices to creep in and make it better. So I'm real excited about what I've learned, especially last week. Now I heard some really great information at the end, but last week I really enjoyed Dr. Efron because that was awesome. That because all that mycorrhizae fungi and like how to find it, like Brain Grow and I had the same idea. Where do you find it?
As far as the soil food web and the actual information on the mycorrhizal relationship with the plants, that is hands down the best guest that we've had to speak on the subject, in my opinion.
You mean last week?
Yeah, last week.
I thought that was awesome too. And both of them, him and Elaine, I love Elaine was funny too. She was like, I could see taking her. You won't sleep in her class. And I wouldn't sleep in Dr. Efron's class either. I was amazed at just the way the information flowed and, and how funny it was when we asked him questions about stuff he wasn't necessarily going to talk about. I love when we do that to them, our guests. We learn so much more that way because they're open to do it. So I yield. That's all I'm doing. I'm just, beating my head against the wall around here and trying to keep up. And Mama's home and she's been feeling real bad the last week or so. So even though we're, not doing too well together, I still kind of care for her. So I'm kind of being a nursemaid, good neighbor right now.
Cool. What about you, Fish Ganja guy? You haven't had a chance to get caught up with you in a little while.
Hold on one second, just move my mic. Okay, sorry, on the headset. Yeah, not too much else is going on at the moment besides the usual growing in soil, painfully slow. See, I am getting close to harvest. I just did a week of winter frost from New Millennium to get the plants all into senescence at the same time. And I'm about to start flush tomorrow. Battle with the root aphids and the vegetative tents. It's going my way, but since I'm using more natural inputs than anything, it's one that it's going to be like a long battle to completely wipe them out. But it is what it is. Other than that,
have you tried nematodes?
Oh yeah, no, they still somehow powered through. So what I'm doing is I talked to Dinesh Optic Foliar about my problem, and he said that ATAC at a 2 ml per gallon, the root dunk, or just drench my plants with my next feeding. He said that'll wipe most of them out, but also get the nematodes too. So I went ahead and did that tonight because I have nematodes already on order. I've got 50 million coming on Monday next week. So I'm going to start a new tea brew with another Metarhizium fungal pack. Basically, it's the fungus pack from Ketolife. I asked them, they said there is Metarhizium in it, even though it's not one of the listed fungi. But it's in there, so I'm going to brew that up, dose it with nematodes and everything next week. And hopefully that should be the end of it. If it's not, I'm just going to burn the whole motherfucker down because I'm done. But yeah, we'll see how it goes.
Yeah, okay. Yeah, so shout out to Optic Foliar. Dinesh was on the show, was a great guest last year, and I'm glad to hear you bring him up. I mean, personally, because I've been reminds me somebody else I need to get a hold of. Something else I need to do is give him a shout. But yeah, that was
Yeah, he's a good guy.
We'll get
Optic
Dinesh.
Foliar.
We'll
Yeah,
get
Optic
Dinesh on again.
Yeah, great product too. From what I had, I haven't got to use it. That's one of the reasons I need to get a hold of him.
Yeah, if you're doing foliar sprays, you really do want to use their product. Transport makes a night and day difference. I'm not a big fan of foliar feeding, but if you do want to do that, it's the product to use to actually make it happen. It's the only product when I worked at Aquaponics Source, the only one that actually showed any kind of difference in nutrient tissue value.
It's also the only product that we know of that can be sprayed on when the lights are on.
No, you can spray. You always should spray. Oh, I'm glad you brought that up. So you should always spray with the lights on.
Oh,
Always.
I thought we had a big talk that night about you shouldn't spray when the okay.
Roger, just to clarify that, it's with oily products. You don't want to spray with the lights on because you'll burn your plants. But if you use
yeah,
it'll be gone within 20 minutes. So it'll be but you usually want to try and.
Steve says no.
So for insecticides, you don't want to spray with the lights on or right at the end. But if you're trying to feed the plant with a foliar, we're getting an echo from someone.
I'll cut mine off. Maybe it's me.
So if you're trying to foliar feed, you actually want to do it an hour after the lights come on, because that's when the stomata, which is the primary means that you're trying to get that into the leaf, are in the largest diameter. But if you use transport, it doesn't matter because you're putting it through the cell wall directly and not having to rely on the stomata.
Correct. I'm just saying that normally without transport, you don't want to use any oily products like neem or, like you're saying, a lot of the pesticides. Whereas most things like kelp or, a lot of the things that we normally foliar our plants aren't going to be super oily and cause them to get sunburned. So those you can normally do with the lights on, which, like you're saying, is when the stomata are open. The only thing from optic foliar that I can say you want to do with the lights off, and this is coming from Dinesh and personal experience, is the switch. Optic foliar switch, you have to use it with transport, but he highly recommends doing that one lights off. Everything else, lights on. But yeah, the other thing that's good, Roger, like I was going to say a little earlier with transport, is it lets you use a lot of those pesticides like Azomax or any of those others at like quarter to half strength, so you can make it go a lot farther for your dollar, because it's getting it into the tissue of the plant systemically, so that way you can actually save a lot of cash and get the same effect.
So transport, is that similar to, now so I know we talked about this a long time, but since we're on it, we might as well, is that, I can't remember, was that something I thought that night, is that possibly like a wetting agent? And is it, when you're saying it's it's
not so much like a wetting agent as it's something that actually opens up the mesophilic layer of the plant and a delivery agent. It delivers the stuff directly into the plant tissue, or the leaf tissue, I should say. So for like that 20 minutes, it opens up the mesophilic layer and lets things transport in. Whereas a sticker spreader, it'll just wrap it around the leaf to the underside where the stomata are. But this, you can just spray directly on the top of the leaf and get the same effect. It's getting into the plant.
Yeah, see, that's another thing. Most of the time on a lot of these products, you don't want to spray the top. You want to spray the bottom, don't you? Or just the leaves. You don't want to spray the bud. We know that. But again, not being a fan of foliar, I've never studied much on it. But Dinesh sold me on the idea that it might be interesting to try, with the products, especially with the whole panel liking the product. You have to kind of trust it then.
Because otherwise you're just feeding your plants one way when you can feed them two ways, and I'm sure they'd appreciate it.
And sorry, the echo, I figured out what the echo was, because I had to turn up to hear fish. And so I had my speakers on too loud.
Well, that's nice that you want to blame me, but you could just own up to it, man. That's cool.
Where did Marty go? I was just about to ask Marty. All right, well, I'm not sure where Marty went. We'll get Marty on if he comes back. What have you been up to, Brain Grow?
Right now, I'm actually trimming up a plant I harvested about a week ago, early Durban. And in the grow, I'm actually coming to a realization that it's no fun to grow all these re-veg plants because they're growing slow as shit. So What I'm probably fixing to do is I was planning on getting it done today, but I had a lot of stuff to do. I was actually working on my chicken coop a little bit today, so I didn't quite get around to it. But I'm going to get all these original skunk number one re-veg plants that I have and stick them all in one grow bed and just go ahead and flower them out where they are right now. And then I'm going to go ahead and pop some more seeds for the purple haze, the super silver haze, and the gold leaf, and get those going as soon as possible. Let's see what else. Other than that, not much else, you know, just doing the normal routine in the garden, feeding the fish and checking pH and making sure my nitrates and stuff are good.
That's awesome. You're starting the ILGM pack, huh?
Yeah, fixing to be starting those pretty soon.
Yeah, I got my juicy pack in the other day. It's had Agent Orange Bud, and Super Lemon Haze in it. So that should be fun. And then I got a cherry pie, and I couldn't pass it up, even though there's so many other strains I wanted. I had to get the Maui Wowie. They're just out. We procured Maui Wowie just a while back, and I heard people talking about it. So just for some giggles, I had to go get it and see how it comes out.
Right on. Another thing that I was kind of messing around with the other day was my mealworms, and I don't really spend a whole lot of time around them. I'll give them a water source like maybe every couple of weeks to once a month. You know, I'm not real tentative to my mealworm farm. And when I checked up on them, they were the biggest I've ever seen them. They're massive, like 2 inches long, like super fat, juicy little things. So I'm getting ready to probably feed some of those to my fish. I'm sure they're going to have a blast.
Awesome. What about you, Mr. Green Jeans? What have you been up to?
But yeah, I've been running the press. Boy, the press is amazing. The rosin press, unbelievable, and it makes incredible medicine. And you know, and I've been eating pieces of it, and you know, trying to figure out how to smoke it. I can't smoke it at all. I have to get a rig. I have to get a special rig, like you said, Steve. And Brain was telling me to. It's impossible to smoke. It makes amazing medicine right away. Just mix it with oil, you know, or or eat it straight. It's incredible. But yeah, it takes a lot of skill. You know, you've got to practice. You've got to work with it and everything like that. And I've only done like 7 or 8 presses, and I'm just barely beginning to get the hang of it. Now I'm getting the lower temperature ones, you know, down 180 degrees and getting a really nice, thick looking, pasty results. And, you know, I think it's really squeezing a lot of the stuff out. It was really interesting squeezing the G13. I don't know if you guys remember, I was talking about the and that was the ones where I got the 11 seeds from 11 plants from 10 seeds. And I was bitching about they didn't have much terpenes. You know, some of them were even really amazingly resiny looking, and they look beautifully dusted and everything, but they don't have very distinctive smells. You know, they're not really strong in the smells department. And it was really interesting because once I pressed a whole bunch of them all together and got to smell the rosin result, I smelled some things that I couldn't really detect there in the original. And it took me a while to put my finger on it. But a couple of other people have agreed that the smell of that rosin is kind of like raw bread dough with a little bit of yeast in it. If you can imagine that. It's a really unusual and odd smell. But really mild, actually, which is really maybe nice. And actually, and again, the taste of the rosin, the taste of the raw rosin from this G13, which I was bitching about, saying, oh man, this, you know, these genetics, they don't have no terpenes, there's no smells in there. But when you eat the raw rosin, it doesn't attack your mouth that bad. It's pretty, it's almost a smoother rosin to eat in raw condition, because none of the rosin is all that smooth to eat in raw condition, actually. It's kind of a little bit rough on the taste buds. You taste everything all boom, and then of course it sticks to your teeth, and you know, and 20 minutes later you have this giant head blast, you know. So it's a pain in the ass to eat. And I'm not recommending that unless you mix it with oil. Oh, and Brain suggested mixing it with the honey was fantastic. But the problem with that is that you want to eat a lot of it. So it's really easy to overdose. You know, already making it a little bit strong to begin with. But here's one of those, one of the regenerated, one of the, this was an AMS. I like that. I had 3 of those AMSs. They were nice. These little
Used to be our partner, Mr. Green. We used to partner with those. AMS was one of our approved seed bank.
Yeah, I thought it stands for the airport. That I thought they told her.
Oh,
maybe I
didn't get. I was doing the wrong thing. I was thinking Amsterdam seed.
Amsterdam Airport. It was the name of the strain, I think. It was they called AMS.
I see.
Yeah, and it was from. I think it's from Greenhouse. I thought it was from Greenhouse. I thought that's where she went. But it was one of the ones they gave me for free, and they're beautiful. You know, it's nice and incredibly resiny. I think really high THC, beautiful, nice structure, pretty good, big yield. You know, good plants overall, really good plants. But all of them, they all suffered. I would say compared to maybe it's just maybe I've just got a collection of plants with ridiculous terpenes, but I feel like here in California we have a lot more smelly plants. I don't know. sounds silly or simplified, but and I have this theory that they over there they
got those
smoke
gassy diesel genetics.
Yeah, they smoke everything over there with tobacco too, and I don't know if you can, maybe they're not appreciating the full flavors that we are over here. Maybe over here it's more common for us to smoke straight flower. No one mixes it with tobacco. So I don't know. Maybe it affects those breeders over there. Smoking too much tobacco. Because it stands to reason that G13 would be perfect for mixing with tobacco. It doesn't have much flavor. It's actually probably potent, has a lot of THC. So I don't know. And the fact that the rosin actually tastes pretty good raw, which is unusual too. It has almost smells like bread, like raw bread dough, which is real interesting. So, yep, got to learn how to smoke that stuff though. I don't know what the heck you do with it. It's incredibly sticky. I tried it in my packs. That kind of works on a really high setting. I put it on the highest setting, and you get tremendous hits with it. But the thing is, it's only smoking part of it, and a lot of it's staying behind. And the stuff that stays behind makes wonderful hand lotion. So, pro tips out there for anyone with packs and is trying to figure out how to smoke rosin. Smoke it down to the last. But Brain was suggesting that actually I'm probably leaving behind some really good THC, which stands to reason. I feel like the early hits that you get are very broad, like you're getting a lot of terpenes to start with, like you're getting, all of the exotic effects of the cannabis first and not so much heavy THC hit. And then as you keep hitting on the packs, eventually it gets around to I think the rosin really resists burning, really resists smoking. You've got to get the super high heat, the dabbing rig like Steve was showing me. I think it's pretty much either the only way. I've heard about the little vaporizers or what do you call them, atomizers, like the Sayonara with the little ceramic donuts, stuff like that. I don't know if anybody's had any experience with that. Steve, you ever seen one of those?
I got a pen I was telling you about. Go ahead, Steve.
No,
there's little ceramic pins. It was interesting. They have little ceramic tops that work all right. It was really interesting. I made a batch of bubble the other day, and I went to press it, and I got no oil out of it, none, just a tiny, enough for maybe one dab. And I thought that was super bizarre. And then I ended up with this really thick pressed hash puck, and so I ended up just taking that, cutting the bag off, and I smoked a piece of it just on top of my weed, like it's someone's feedback. But I smoked a little bit of that on my on my weed, and it was just the stoniest high. It was just like smoking some old school hash. It was really good. It was different, but no oil in it. I couldn't believe that it was 3 days dried. It should have been totally juicy and just nothing. I'm still kind of baffled on that. But I also don't know how old the trim was or anything. It was just some trim a friend of mine gave me. He had laying around, so.
It's amazing the different genetics too. Now that I was sticking to the one, I had the one big jar with a couple of ounces when I was trying to learn, and it was all the same. And now and then I tried a few presses of a couple of different things, and it was like, wow, completely different. This one thing I didn't expect so much came out of it. And I guess, in retrospect, but wow. It wasn't a plant that I would have suggested. If you'd asked me, does it have a high THC content? I think I would have guessed not, that it was low. But the rosin presses out of it seems to be, and I don't know what the THC content of that rosin is, but it pressed a whole ton, and it looks fantastic. And smells and tastes great. So,
Well, that wad of resin you showed us last week, that was pretty impressive for your first time on your homemade rig. That was a big ring of resin compared to what I've seen with a lot of other folks, what they're putting in there. So that was pretty awesome.
I probably put a lot that, trying to figure out how much to put in to start with is definitely the big thing. And I don't even have any bags yet. And the bags are really good, not just... You actually don't really even need the filtration that much. It's pressing out, and the way I'm using it now, the parchment paper, it's pressing out, and it's easy to collect. But the thing is that I think the bags help to contain if you want to use a large amount, like maybe over 7 or 8 or 9, 10 grams on a 3x5 plate. Then maybe the bags actually act more almost as a containment kind of thing. So it prevents it from squishing out any more, and it holds the puck to that size, right? Whereas now, if I put that much in, it's going to squish all the way out to the edges of the plate at a certain point, a certain amount. It's just going to squish right out to the edge of the plate if there's no bag.
Bags help a lot with just separating the plant material. You get much cleaner material. I use bags pretty much every time.
That as well, but I'm saying that doesn't seem to be a problem unless you maybe press twice or fold over the puck or something, which is a cool, you can get a little more out of it. Seems like the plant material, there isn't, the oil just squeezes right to the outside edge, and there isn't really any in it so far. I mean, I don't know. Maybe with other buds, it's going to be completely different.
That's pretty much how I've been doing it too, Green Jeans. I haven't had much plant material in mine unless I've grinded it beforehand.
Throw it in. Yeah, don't grind it. Throw it in a coconut oil, your pucks, and then cook them down. You'll get a shit ton of good CBN.
Yeah.
So,
I made some with the isopropyl that made topical, and I didn't realize that all of these were made with my White Wizard, which is basically remixed White Widow, and I kind of had a suspicion that it had a strong sort of a pain-killing effect, and we found out when you made it into the topical, man, and you rub it on your hand, on your skin. That's when you can really tell. I mean, I got it also from eating a piece, and I got the entire head numbed, like as if I'd eaten Novocaine or something like that. But the topical is like, wow, you can totally feel it. So it's definitely sorry, yeah.
Let's switch to a Hogmaster here. Looks like he's got some.
He's showing us some oil.
Just finished it with a bunch of pucks and used a bunch of resin, or the rosin pucks from the keef that I did.
Awesome.
Those actually cook really, really well. The pre-made bags of keef.
Awesome. Cool. You got your camera still on, if you're aware.
Oh. You can hold it up to the sunshine and not even.
Cool.
By the way, while we had a little quick space there, Marty did put in the chat that he would be right back a while ago.
Yeah, that was a little earlier.
Yeah, but it was right before he left, so I didn't know, and I didn't know if when Hogmaster's done, we got anybody in chat tonight?
I was fixing to say, speaking of chat, who do we got live tonight?
Got a whole ton of people earlier.
Thanks for sharing, Hog.
Yep. Sorry, I didn't mean to cut you off, Mr. Green Jeans. I just wanted to. He was holding up the thing you were talking about. So I muted you for a second because it was feeding back.
Also, I'm take off, guys. Have a good night. It was great hanging out, and Steve, I'll talk to you soon.
Cheers.
Later, fish.
Later, fish.
Alrighty. I'll have some. Hopefully, I'll have some big announcements next week. I got some stuff in the works that I can't talk about quite yet, so we'll see if that comes to fruition or if we have to wait longer. And yeah, I'll be at the Science of Regenerative Organic Chemist Conference all weekend. If anyone's looking and wants to come talk and hang out. And what else we got going on? Do Grows Cubs coming up on the 15th. So we'll get on that. And yeah, we got some really awesome guests lined up too in the future. So, anybody else have anything before we close out? I'm going to end a little bit early. I'm in Colorado right now on a business trip, so I got to get to one other thing later this evening. But does anyone else have anything?
I guess we're good.
Awesome. Alrighty, well, we'll be sorry for a little bit of early start this week. We had it was just to help facilitate a little more time with Elaine, so we'll be back normal time next week. And thanks a lot for everyone joining us.
Thanks all the live viewers.
Yeah,
keep on growing.
Thanks a lot for Dr. Ingham for coming on the show as well. We've been talking about having her on for a while.
Dr. Elaine.
All right, thanks everybody. Take care.